Antibody is a chemical substance. Antibody is a protective. Antibody is a protective chemical substance. Antibody is a protective chemical substance. So it is produced. When it is produced, so antibody is a protective chemical substance. It is produced when the antigen or foreign substance, entered into the body. So this is called antibody. Antibody is a protective chemical substance. It is developed or produced when the antigen, this antigen is nothing but foreign substance. The foreign substance is called antigen. So this foreign substance entered into body. So then it produces, antibodies are produced. Then the antibodies are produced. So protective chemical substances. These are produced when antigen or foreign substances entered into the body. Thus, this is the definition for antibody. So it means the antibodies are produced in response to the antibodies are produced in response to the presence of antigen. Presence of antigen. It's called antibodies. In previous classes, I have discussed in depth structure of antibody. Antibody is generally is a protein molecule, protective protein or chemical molecule. Generally antibodies are Y-shaped molecule. And they are protein molecules. Antibodies are generally protein molecules. Chemical antibodies are chemical, that is protein molecule. And they have Y-shaped structure, and they are made up of 4 polypeptide chains, 2 heavy chains, 2 light chains. That's why antibody is represented as H2N2. I have discussed in detail the structure of antibody. Antibody consists of 2 regions, variable region and constant region. So these 2 polypeptide chains are end together by disulfide bond. This is the structure of the antibody. Clear? So antibodies for protective chemical substances are protein molecules. They are produced in response to the presence of antigen in the body. So these antibodies are produced by, we know already, the antibodies are produced by B lymphocytes. B lymphocytes. Antibodies are produced by B lymphocytes, and they circulate through blood and lymph. They circulate through the blood and lymph. In our body, so different millions antibodies are produced. And these different millions of antibodies are recognized the different antigens, specific antigens. Clear? So this is the definition for antibody. Antibodies we know already. Antibodies are produced from B lymphocytes. After the production, they are circulated through blood and lymph. Different millions antibodies are produced in our body, and these different millions of antibodies are responsible for recognize the different specific antigens. The antibodies recognize the different specific antigens. Okay. So there are 5 major classes of serum immunoglobulins antibodies are present. So how many types of antibodies are present? Very important. So different million antibodies are produced in our body, of course. Among these, there are 5 important major serum immunoglobulins are produced in our body. So next I will discuss about types of antibodies. Types of antibodies. Major types of antibodies. Types of immunoglobulin antibodies. Types of immunoglobulin antibodies. Types of immunoglobulin antibodies. So, in our body, there are 5 major serum immunoglobulin antibodies are produced. There are 5 major serum immunoglobulin antibodies are produced. They are IgG, IgA, IgM, IgD and IgE. These are the major 5 serum immunoglobulin antibodies produced in our body. I have discussed one by one. First one, IgG. First one, IgG. Immunoglobulin antibody. It constitutes IgG. It constitutes about 70 to 75% of total serum immunoglobulin antibody. So that is IgG is the major and very important antibody produced in our body because it constitutes about 70 to 75% of total serum immunoglobulin antibody. It is major and very important antibody because it fight against bacterial and viral infections. Bacterial and viral infections. Bacterial and viral infections. Yeah? And it is important and major antibody produced in our body. It constitutes about total 70 to 75% of total serum immunoglobulin antibody. It's very important because IgG is an antibody that fight against the bacterial and viral infections. Second one, IgA antibody. IgA constitutes about 10% of total 10% of total It constitutes about 10% of total serum immunoglobulin. Globulin antibody. It is also IgA. This antibody is also very important because it is responsible for local immunity. It is responsible for local immunity. It creates or is responsible for the local immunity. And it also IgA antibody is also attacks bacteria and viruses. Bacteria and viruses. So IgA antibody also attacks bacteria and viruses or fight against bacteria and viruses. It is responsible for local immunity. It constitutes about 10% of total serum immunoglobulin antibody. Next, third one, IgM. It constitutes about 5 to 10%. IgM antibody constitutes about 5 to 10% of total serum immunoglobulin antibody. It is also referred as macroglobulin. It is also referred as macroglobulin because of its large size. Because of its large size. This is about IgM. It constitutes about 5 to 10% of total serum immunoglobulin antibody. It is also referred as macroglobulin. IgM is also referred as macro, means large or big because of its larger size. Next, fourth one is IgD. Fourth one is IgD. So it constitutes about less than 1%. IgD antibody constitute about less than 1% of total serum immunoglobulin antibody. So IgD, this antibody is present in the intestinal fluids. It is present in the intestinal fluids. IgD is present, this antibody is present in the intestinal fluids. Last, fifth one, IgE antibody. It also constitutes about less than 1%. It also constitutes less than 1%. And it is responsible for, or these IgE antibodies are produced at the time of allergic reactions. They are produced at the time of allergic reactions. So these are the 5 major and very important antibodies produced in our body. So in our body, millions different antibodies are produced because among these These are the 5 major and very important antibodies produced in our body. So IgG, IgA, IgM, IgD and IgE. These are the 5 major and important antibodies produced in our body. Clear? So antibody is a chemical substance or a chemical molecule. It is generally antibodies are generally Y-shaped, so molecules. Okay. These antibodies are produced in the response in our body in the response to the presence of foreign substance called antigen. So antibodies are generally produced by B lymphocytes and circulate through blood and lymph, and these are the major and important types of immunoglobulin antibodies. Clear? Next concept. Next comes active immunity and passive immunity. Active and passive immunity. Active immunity and passive immunity. And passive immunity. I have discussed one by one. What is the active immunity and what is the passive immunity? So, basically, immunity. So, basically, the immunity is classified into 2 types: active immunity and passive immunity. So, I will discuss one by one what is active immunity and what is passive immunity. First one, active immunity. I will discuss about active immunity. Active immunity is the immunity. Active immunity is the immunity. It is developed. It is developed. It is developed when the individual is exposed to, when the individual is exposed to the antigen, by way of infection, it is called active immunity. So active immunity is the immunity. It is developed. How exactly the active immunity is generated in our body? So active immunity is the immunity. It is developed when the individual is exposed to the antigen. The individual or body is exposed to the antigen by the way of infection. It is called active immunity. So active immunity increases resistance. Active immunity increases the resistance against subsequent infections for particular microorganisms. So the active immunity increases resistance against subsequent infections for particular microorganisms. So the antibodies produced when the antigen entered into our body. Here, antigen or foreign substances or pathogens entered into our body. Immediately antibodies are produced. Antibodies are produced from our B lymphocytes. Immediately B lymphocytes produces antibodies. So the production of for first infection or for the first time of infection, during first infection, the production of antibodies is slow. For the first time of infection, the production of antibodies is slow, and it requires several days. The production of antibodies is slow, and the production of antibodies requires several days. It means the production of antibodies requires several days during the first time of infection. For the first time of infection, production of antibodies is slow and it takes several days. Clear? So next step is for the subsequent infection of same microbe. For the subsequent infection by same microbe, the antibodies are produced immediately. And enhances the resistance against the infection. See, for the first time, got the point? For the first time, production of antibodies for the first time of infection. Production of antibodies is slow and it requires several days. But for the subsequent infection of the same microbe, the antibodies are immediately produced and enhance the resistance against the microbe or pathogen or infection. This is because in subsequent infection of same microbe, the antibodies are immediately produced within a short time. The antibodies are produced during subsequent infection because for the first time of infection, during first infection, the memory T cells are produced. Memory T cells are produced. So these memory T cells can provide long time immunity. Produced a long time. This is the reason. Okay? So in second or subsequent or second infection, second, third, fourth, or in subsequent infection, so immediately the antibodies are produced, but not in first infection, because during the first infection, memory T cells are produced. That's why these memory T cells produces long time immunity. Here, that's why. So in the second time or subsequent infection, antibodies are immediately produced because memory T cells are produced. These memory cells, T cells are memorized. The microbe, it infects previously and produces long-time immunity. This is about the active immunity. This is about that. So active immunity. Clear? So then next one, active immunity. Next is passive immunity. Next, I will discuss passive immunity. Passive immunity. Next, I will discuss about passive immunity. We discussed about passive immunity. Passive immunity is also naturally developed. Passive immunity is also naturally developed. Passive immunity is naturally developed. Passive immunity is naturally developed when a non-immune, individual receive the antibodies from other individual, is called passive immunity. Listen clearly. Passive immunity is also naturally developed. Passive immunity also naturally developed. When a non-immune individual is in some individuals, the resistance power or immunity power is not developed. So that persons are called non-immune, means there is no development of resistance power or immunity, called non-immune individual receives antibodies from other individual or donor is called passive immunity. For example, anti-serum from animal origin. Anti-serum from animal origin. This is the best example. See, you got the point. Passive immunity. So the non-immune individuals can receive antibodies from other individuals. So best example of anti-serum from animal origin. Next, so then passive immunity are of two types. Passive immunity are of two types. Passive immunity are of two types. Passive immunity. Passive immunity are of two types. First one, two types. Passive immunity are of two types. They are, first one, natural passive immunity, and second one, artificial passive immunity. Artificial passive immunity. So there are two types. There are two types. There are two types in passive immunity: natural passive immunity and artificial passive immunity. I first discussed about natural passive immunity. First, I have discussed about natural passive immunity. So, the infants receives the infants receives immunity from their parents by two methods. The infants receive in natural passive immunity. The infants receives immunity from their parents by two methods. First method, through breast milk, through antibodies, through placenta, through breast milk. By these two methods, the infants receives the immunity from their parents. So, through breast milk, first one, A, through breast milk. So, the initial milk secreted, the initial milk colostrum, secreted during initial few days of lactation. Few days of lactation contains the antibody is IgG. Contains the antibody IgG. Contains the antibody IgG. So this antibody produced are present in the colostrum. So during the initial milk is called colostrum. It is secreted during initial few days of lactation. This colostrum contains the antibody IgG. Okay. Then the infant's intestine. Is porous in nature. So, the intestine of infant is porous. That's why the antibodies IgG, IgA, these antibodies are directly absorbed. These antibodies directly absorbed. These antibodies are directly absorbed. So then, after the one age, after 1 year, after 1 year, the pores closed. After the age of 1 year, the small pores present in the intestine of infants are closed. Because of closing of the pores, the antibodies are not absorbed. Infant's intestine, the pores are closed after 1 year age. So then the antibodies are not absorbed because of closing of pores. Then the antibodies are not absorbed. The antibodies may coat. Then the antibodies are not absorbed. Then the antibodies are coat to alimentary canal. Coat to alimentary canal. So they form, the antibodies form the coat around the alimentary canal of the infant and escapes from the infection. And through the infants also, acquire or receive immunity through antibodies transferred through placenta, and give or provide natural passive immunity. See, the natural passive immunity. In this, the infants receive the immunity by their parents by two methods. First one through breast milk or breast milk, and second one is through placenta. Clear? So this is about natural passive immunity. So breast milk, the initial few days, the mother secretes the milk. It is called colostrum. It is rich in antibodies. For example, IgG and IgA antibodies are rich in colostrum. So these antibodies are directly absorbed by the infant's intestine because the infant's intestine contains pores. Through these pores, the antibodies are directly absorbed. After the age of 1 year, these pores are closed, and because of this, the pores are closed. Because of this, the antibodies are not absorbed. But the antibodies after the 1 year age, they form the coat around alimentary canal and escapes from the infection. Then, second one, the infants also receives the immunity by placenta or through placenta. So the through placenta, so the antibodies passes into the fetus through placenta. The antibodies are entered into the fetus, then provides the natural passive immunity. This is about natural passive immunity. Second one is artificial. Second one, artificial. Passive immunity. Artificial passive immunity. So artificial passive immunity is one in which the immunity is developed. in which the immunity is developed when the antibodies are administered artificially. So developed when the antibodies are administered artificially from donor, it's called artificial passive immunity. It's developed when the antibodies are administered artificially from the donor. It's called artificial passive immunity. So the antibodies produced in the other vertebrates like horse and cow. These antibodies are produced in the other vertebrates like horse and cow. These antibodies produced in the horse and cow are isolated and injected into when they are required. This is when they are required. This is an artificial passive immunity developed when the antibodies are administered artificially from the donor. For example, these antibodies are produced in the other vertebrates like horse and cow. These antibodies produced in the horse and cow are isolated and injected into the other individuals when they are required. For example, antivenin is an antibody. It is injected at the time of a snake bite. For the encounter of snake venom. For example, antivenin is an antibody. So it is used against the snake bite for encounter of snake venom. So it's called artificial passive immunity. Natural passive immunity and artificial passive immunity. Then next, colostrum. Note colostrum. What is colostrum? Colostrum is a yellow colored. Colostrum is a yellow colored milk. Colostrum is a yellow colored milk produced during initial few days. Of lactation. Initial few days of lactation is called colostrum. is called colostrum. See, colostrum is a yellow colored milk produced during initial few days of lactation is called colostrum. Colostrum is rich in protein. It is rich in a protein called lacto-albumin. Rich in a protein called lacto-albumin and immunoglobulins. IgA, and low fats, sugars, minerals, vitamins, and also contains enzymes for efficient digestion. This is about colostrum and composition of colostrum. Colostrum is a yellow colored milk. It is produced during initial few days of lactation called colostrum. So colostrum contains a protein called lacto-albumin, immunoglobulin IgA, low fats, sugars, minerals, vitamins, and enzymes are present. Enzymes are required for efficient digestion. So, this is about colostrum. So next, difference between innate immunity. Difference between innate immunity and acquired immunity. Difference between innate immunity and acquired immunity. Innate immunity is a non-pathogen. It is a non-pathogen specific type of defense mechanism. It is a non-pathogen specific type of defense mechanism. Next, it is inherited from parents. Since birth. So innate immunity is transferred or inherited from their parents since birth. That's why it is called innate immunity. So inborn immunity. Next, third one, innate immunity operates by providing barriers. When the microorganisms are introduced. See, the innate immunity operates by providing the barriers when the microorganisms are entered. We know that the barriers of so innate immunity, physical barriers, physiological barriers, then cytokine barriers, cellular barriers. So then acquired immunity. Acquired immunity. Acquired immunity. So it is a pathogen specific. It is a pathogen specific type of. It is a pathogen specific type of defense mechanism. So then it is acquired after the birth. 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 Next one, it operates by providing primary and secondary responses. Primary and secondary responses mediated by B and T lymphocytes. Mediated by B and T lymphocytes. This is the difference between acquired immunity and innate immunity. Acquired and innate immunity. Then last one, it has not a specific memory. It has not a specific memory. It has immunological. It has immunological memory. It has immunological memory. This is the difference between innate immunity and